I want to welcome everyone who is in attendance, or for those who are listening to this at a later time on the recording. Thank you for joining us tonight, and we hope that between today and our next session in June, that everyone is able to take away a few pieces of information about the microbiome. So today we want to cover just the basics. We want to focus really on what the microbiome is. Why is it relevant to swine production? More importantly, how do we measure it? How is it reported? And also, what are the next steps? So as we move forward, one of the first things we want to cover tonight is what is the microbiome? The microbiome is really a collection of microbes that create a community. And this community can be both in the environment as well as within a host, an animal, or some other type of living organism, insect, fish, etc. When we think about the microbiome itself, it is not just bacteria. And many of us want to associate the microbiome as just that, as just bacteria. But really, a microbiome is a series or collection of microbes, and that could be viruses, fungi, and bacteria. We also know that about 90% of human health issues can be related back to the gut and the microbiome itself. And so, again, I think this is something that many of us don't think about. Certainly when we have gastrointestinal disorders or something of that nature, or maybe ear infections, we might think of the microbiome there. But the reality of it is you'll see here in a little bit, that microbiome really has a vast function beyond just the space in which it is located. We know that the microbiome itself, if we look at humans, just in the intestine alone has about 10 million genes. And so there's a phenomenal amount of DNA that's associated with this. So as I mentioned, the microbiome, we think about that typically in the gastrointestinal tract, but the reality of it is the microbiome is present everywhere. We look at the microbiome in the skin, within the gut, the respiratory tract, the mouth, the reproductive tract, even within our eyes and our ears, and more recently, we're even finding it within the own bloodstream. And so the microbiome is not just in one location. And again, many of us want to put it mainly in the gut. One of the other questions that comes up quite regularly is, what's the difference between the microbiome and the biofilm? And I want to share, just take a moment and talk to you about the two differences and then how they are, in fact, both integral to what we're talking about tonight. So in simplistic forms, I drew a version of a biofilm on there. It is a group of organisms that are living in some environment. Again, it does not have to be within an animal. This environment could actually be within a water line, as most of us might be thinking about a pig barn. It might be on the surface of the farrowing stall or on the flooring itself. But basically what we're looking for is a few organisms are sitting next to each other, and through a very serious set of signaling, they start to pull together into their community. As these organisms pull into their community, they start to create this matrix on the outside of that community. And so one of the things I kind of think about is the image of a biosphere, and the big glass bubble sitting on a piece of land, and inside that bubble are all the people that live within the biosphere. That's exactly what the microbes are doing in the biofilm. They are creating this house, if you will, and that house is comprised of sugars that are very similar to some of what our own bodies express. And so the immune system is not able to detect those organisms when they're encased in this matrix. This matrix, though, is not solid. And so this matrix does allow for fluid to move in and out of that location. It allows for, of course, waste products from those microbes to leave their little housing, if you will, and it allows for new nutrients to enter. In addition to this, what we also do suspect, and we have been able to document it, is it allows for the ability for antibiotic resistance to occur. Because if we are by chance using an antibiotic to try to control these organisms, they can kind of dissimulate that antibiotic, if you will, at a low enough level that it does not become toxic, but it allows them, of course, to genetically select for organisms that are resistant. Eventually, that housing breaks apart and organisms are released. And so in this bottom image here, you'll see these two darker color organisms leaving. And so in this case, maybe those are the two that are antibiotic resistant, and they are now leaving that biofilm. The biofilm will completely dissolve, and the organisms will shuttle on to different locations and create new biofilms. And so this cycle just continues to repeat within the host. We see this in things like urinary infections in the bladder. E. coli has been known to create these biofilms. We see them in children's ear infections. We see them in people with lung disease like chronic pulmonary disorder. And so this is nothing that is novel or new, but it is kind of the process of what a biofilm looks like. And so I took a small image, oops, my apologies. I took a small image of a video here of what a biofilm looks like. And so this is Pseudomonas aeruginosa, and that is a biofilm. And so what you're seeing are the mounds, if you will, those little biospheres, and all the little green rods in there are, of course, the organism itself. And so the matrix is holding all those organisms in a plume, if you will, on that slide. And each one of those then is its own community, its own biofilm. And so over time, then that biofilm will break apart, those organisms will leave, and it will move on to other places. So why am I doing this? Why am I spending time on biofilms? Well, it's because biofilms and microbiomes are very much similar, and they also work together. And so when we are thinking about microbiomes, there are actually biofilms within microbiomes. And these biofilms aren't just always single community organisms, right? I just showed you Pseudomonas, but sometimes there can be multiple organisms or species within those biofilms that interact well with each other. And so this is a very, very complex system when we start talking about the microbiome. And that's why I wanted to bring this in, was because biofilms and microbiomes, while they behave differently, also are interacting together within the host. So let's talk a little bit about the microbiome influence. The microbiomes actually have been shown to have both potential impacts on short-term and long-term illnesses. And we do know that disturbances with the microbiome can cause human health issues. Some of the issues that are related to microbiome dysbiosis will be things like pulmonary disease, liver inflammation, ovarian dysfunction, metabolic disorders, and even cognitive decline can be associated with microbiome dysbiosis. How this happens is actually quite interesting. And so if we look at just the gut microbiome and we exclude skin or eyes or any of that at the moment and just look at the gut microbiome is not just only there to help digest feed particles as they are passed through, but the microbes themselves will produce metabolites, and these metabolites will actually signal the receptors on the host. So we call them toll-like receptors or GPRs. And again, this gets very complicated, so we're not going to spend a large amount of time on it, but these metabolites can, to your left, influence the intestinal epithelial itself and what happens at the intestinal level. So it can change metabolic programming, it can increase immune activation and do some of those types of functions. But interestingly enough, those metabolites can also be passed into the bloodstream and be moved to other parts of the body. And so in this situation, the gut microbiome is producing these metabolites, and these metabolites are going into the bone in this situation, and they are triggering things like bone marrow development. They're also, of course, increasing things like cholesterol metabolism and glycolysis. And so that's where the gut microbiome becomes very interesting very quickly, because it is not just within the gut that it stays. It can, or the metabolites themselves can move out of the gut and have this greater impact on the body. And so really, just to kind of reiterate that picture, what we see are the metabolites trigger the toll-like receptors on the host. That follows through with T and B cell recruitment, so some immune function activity. The goblet cells within the gut are also upregulated, and we see more mucosal associated microbiome function, or we see short chain fatty acid production being increased. And then that short chain fatty acid product can also again go into the body and have additional influences, not just on the immune system, but on other parts of the systems. And so when we look at the gut, if we think about the main function of the gut, most of us would think about nutrient digestion and absorption, and that's exactly what we would think of with the microbiome. Breaks down carbohydrates, breaks down proteins into amino acids. It's involved with fatty acid production, cholesterol, vitamin synthesis with some of our microbes. And that's where it really stops from the simplistic standpoint. All these other things are actually very intriguing. We get GABA synthesis with the microbiome. We get anti-inflammatory functions. We get anti-carcinogenic functions outside of the gut, all associated with what's happening with the gut microbiome. In addition to that, the short chain fatty acids are going into the brain, and we're seeing things like brain signaling changes. And so depression and some of maybe those other types of brain dysfunctions can be influenced by the microbiome itself. So I'm going to move briefly into just reminding you some classifications of organisms before I start talking about the organisms that we are focused on. When we classify organisms, we start with domain and we work down to species. When we start talking next series on the actual organisms, we're going to spend a lot of time talking about genus and species. That tends to be where most of the papers are reporting at that level in terms of what's present in the microbiomes. So again, we're just going to be focusing on the genus and the species. I'm going to back up for a moment, though. We do talk some about the phylum, or the phyla in this case. The four most common phyla within the gut of the animal or in the human would be these four on your left: Firmicutes, Bacteroides, Actinobacteria, and Proteobacteria. Within those would be some of maybe the organisms you're more familiar with. So Lactobacillus and Clostridium is part of the Firmicutes. E. coli or Escherichia is part of the Proteobacteria. And so again, not all organisms are beneficial, some are pathogenic, but this is what we're typically looking at for classifications. When we start thinking about the microbiome within the animal, as I mentioned, there's different parts of the animal and they have different parts of different microbiomes. Within those microbiomes, there's different communities. And this slide really just demonstrates what some of the more prominent organisms are within those different communities, if you will. So in the mouth of the animal, we see more Firmicutes and Bacteroides and Proteobacter. When we get into respiratory, we start to see some Actinobacteria in there. The gut, Lactobacillus is going to be a common one that most of us would be familiar with, as well as Enterobacteria. And then reproductive, we see a lot of Firmicute organisms in there. So again, as we think about these different communities, we need to recognize that they don't all look the same. Right? They all have different functions, and there's different needs for why those organisms are there. So when we start taking that phyla, those four classifications within phyla, and we look at them in general, right? Look at that part of the classification. These would be the more common organisms that we see within the gut. These are going to be the ones that when we talk next time, we're probably going to see more of. The most prominent one within the gut is the first one, is the Bacteroides family. So within the gut itself, again, it's not all the same community from stomach to colon. We actually do see that within the gut, we see different fractions or different communities set up within those microbiomes. This is a very simplistic diagram that I'm putting up to show you a couple of different organisms that are present within the different locations of the pig. And again, these are going to change from time to time. But what I'm trying to stress upon showing you these slides is to get you to be thinking about when you are evaluating microbiome, you need to understand where you're looking and what you might be looking for. And knowing that if you're sampling in one area, it doesn't mean that something is the same in a different area. The other piece to this, when we think about the microbiome, is really understanding that taking a fecal sample from a pig does not mean that that's what's actually in the lumen of the intestine. So we do know that the microbiota of the feces is about 0.38 and 0.75 similar to the lumen of the small and large intestine, respectively. So what we're collecting in the feces does not exactly represent what's happening within the pig. We also know that, of course, based on the function of the parts of the intestine, we're going to see different types of organisms showing up. One of the things that I think is really interesting is that it's the small intestine organisms, so those that are present in the jejunum and the ileum, they tend to have a greater impact on host immune function than those in the larger intestine, but the large intestine have greater diversity in terms of the organisms present. And so with that, we can kind of, again, think about not just the location within the gut, but the layer within the gut, if you will. So at the luminal level of the gut, we see microbes that are more interactive with feed particles and responsible for digestion. But when we go into the mucosal layer of the gut, those organisms are more likely to interact directly with the intestinal cells. They're more likely to be influenced by dietary function, and they can attach the mucin for multiplication. And so again, we're kind of narrowing this down, right? The microbiome is different in different parts of the animal. It's different within different parts of the small intestine and the large intestine. And it's even different in those pieces of intestine in terms of what's at the lumen level versus what's in the mucosal layer. So how does this all happen, right? As we're being developed as a fetus, we're sterile, right? The environment is sterile. And in fact, what they're finding now is in utero, there are actually microbes present in the amniotic fluid. But those microbes are not necessarily colonizing the fetus, but the metabolites themselves that those microbes are producing are involved in programming, signaling functions that are happening within the fetus. So again, this is quite interesting, right? Because again, we want to say that the fluid is sterile, but fetal programming or microbial programming actually is starting at the fetal level before the animal is ever born. Once an animal is born, they'll pass through the vagina, they'll pick up whatever microflora is in the vagina. We also now see that the mom has this pathway called the enteromammary pathway, where her immune cells are grabbing organisms out of the intestine and transporting those live organisms into the milk that she's excreting through the mammary gland. And that's, of course, populating that small and large intestine of the offspring. And then over time, those offspring are picking things up through oral exposures as well as fecal-oral transfers. And what we'll cover next time is more of this conversation around how's this happening, why is this happening, and then what changes can happen to influence it. And so we'll talk a bit about diet and sickness and even the use of antibiotics and how that changes in the pig over time. But this is just a quick example of some things to consider. For example, when we feed a diet high in resistant starch, we're going to see more ruminococci present in the intestine. And Amy Petrie was able to demonstrate that high fiber diets with xylanase changed the population within the gut. And so these are just two quick examples, but we'll talk more about that at our next webinar. The last thing I want to cover before I turn it over to Dalton is the starting process on now we know that this is all different across the animal, but how do we collect? And so as I mentioned, fecal samples do not equal what's in the lumen and they do not equal what's in the intestinal mucosa. And so you need to be very aware of what you're trying to analyze and what you're looking for specifically to ensure that you're collecting the right sample. In addition to that, you need to be aware that some of these microbes are anaerobic, and we want to make sure that we're working in an anaerobic situation versus aerobic. And we need to minimize the time that these microflora are outside the body before they're frozen in time, if you will, or the DNA is extracted to get a better feel for what's actually in that sample. Because the longer we leave them out, the more likely that microflora is to change, and it's no longer going to be representative of what was actually in the animal. So as I mentioned, we're going to collect them very quickly. We may choose not to put an additive in. If that's the case, we can potentially store at room temperature for 24 hours and then put it in a -20. Or if we want to do an additive collection, we can put something in like a 95% ethanol or an RNA later and do an extraction at a later time. So with that, I'm going to turn this over to Dalton, and he's going to talk a little bit more about how you analyze the data now that you've collected it.
Yeah, so now that Dr. Greiner has gave a good overview of what the microbiome is and what it may influence in the body, why it's important, I'm going to spend some time talking about how we look at the data, how we sequence the data, and what it really means at the end of the day. So I'm going to start with just talking about some general considerations when we think about conducting studies designed to look at microbiome differences. And so generally, we're still going to follow all the rules of thumbs of good experimental design and everything we would normally think of in any other trial. But there are some things that I wanted to point out that I think are a little more unique in terms of microbiome trials. And so the first thing I wanted to point out is power analysis relating to microbiome studies. And so typically we would do a power analysis to determine the number of samples or experimental units we need to pick up what sort of statistical difference we think is important in a trial. In the case of microbiome studies, though, those methods are not valid and it's hard to determine an effect size and those sorts of things. And so we really don't conduct and can't conduct power analysis at this time for microbiome studies. And so kind of our general rule of thumb is survey the literature and look for studies that have successfully been done in either your species of interest, sample type, and then kind of go from there to build off your study. Another discussion that's often had is the concept of pooling samples. So, for example, if we're in a pig barn and we have a pen of pigs, that's our experimental unit, should we pool a fecal sample from all the pigs into one sample, or should we sample 1 or 2 pigs individually? And so there's some pros and cons to those approaches. And so for pooling samples, there is the benefit that you're likely reducing the number of samples you are going to take, and then on the back end cost associated with sequencing. It may, at the end of the day, more accurately represent the average of your experimental unit or a pen in most cases with pigs. But with that, you have to acknowledge that you're likely going to lose sensitivity and also diversity of the sample, as things that maybe are, or microbes that are rare in certain samples may get hidden by the more common microbes. Additionally, metadata are extremely important in this case, and metadata being just all the extra information associated with your study and your animals you're sampling. And so it's important to collect all the possible info that may have an impact on your results. And so things outside of just treatments, right? So what farm you sampled from, the sex of that pig, the age of that pig, and those sorts of things. And then generally, last thing, just consistency is really key here, right? And so making sure that you're staying consistent in your methods, your sampling collection, storage times, and that sort of thing, to make sure that you're not causing any unintended consequences to the results due to things outside of your variables of interest. So now I'm going to go into a little bit about the sequencing approaches that we see and what we can do with microbiome samples. And so traditionally, we would see microbiome or microbial studies using culture-dependent techniques where we're identifying microbes in samples based on cultures in the lab. The issue with that being that it limits identification to only microbes that can be cultured. And it's been estimated that actually only about 1% of bacteria can be successfully cultured in the lab. And so when we think about the diversity of bacteria that we're interested in and those complexities, it really limits us there. And so as we've advanced technologies and with the advent of culture-independent technologies, so next generation sequencing technology coupled with bioinformatics, that's allowed us to move towards what we currently see today with microbiome research in characterizing some of the more complex communities associated that we're interested in. So just briefly to touch on some of the next-gen sequencing technologies, and this is more just to expose everybody to some of the names of these equipment, because oftentimes at our DNA facility, if we wanted to go submit samples, you need to know what equipment you're requesting to be used. And so this is just kind of based on your question of interest and what you need, some of those options out there. And so if we're interested in using short read sequencing, we would be looking at things like the Illumina MiSeq, HiSeq, or the NextSeq. We're currently using mostly the MiSeq right now, and those are the low cost and low error rate technologies available. But if we need to use an approach that requires more long read sequencing, so up to around 20 kilobase pairs, we're going to have to use different equipment in that case. So an Oxford Nanopore or a PacBio sequencer, those are lower throughput and higher cost technologies, but allow more long read sequencing. So really at the end of the day, when we're going through microbiome studies and looking at some of the communities, we're really interested in a couple questions in my mind. And I think this diagram that I've adapted from Dr. Schmitzesser here does a good job of addressing those. And so kind of at the basic sense, we're looking at the community structure as a whole, meaning who's there, what microbes are there. And so you'll see underneath all of these, I've highlighted some of the approaches. I'll go into a little detail about those, so I'm not going to touch on them now. From there, we could be interested in a little more further detail in terms of the genetic potential. So what can they do in terms of function? And then kind of at the lowest level approach, or the most detailed gene expression data. So what are they actually functionally doing in that host? So to answer the who is there question, we're going to use what's called amplicon sequencing or rRNA sequencing, where we're using a specific marker gene of interest to sequence, and then from that align and determine what microbes are present based on those marker genes. The most common of these marker genes that you'll hear about and read about in literature is 16S. So 16S rRNA sequencing would be an amplicon sequencing approach. This approach allows us to determine relative abundance differences in microbes present in a community, and then compare those abundances between groups. So if we're running a trial with different treatments, what are the differences in individual bacteria present in those groups? But with that, we are limited in terms of resolution, where we really can only look at the genus and species level resolution there. And then also it's difficult to determine differences or distinguish organisms with very similar sequences. And so if whatever marker you're using, 16S, for example, if two different species of bacteria have a very similar sequence in that gene, we can't necessarily distinguish between those organisms. But at the end of the day, amplicon sequencing or 16S sequencing allows for high throughput. And so we can sample and sequence a lot of samples, and it's very cost effective. So we're looking at $10 to $50 a sample, and it does a good job of providing a high level view of the microbial community structure in those organisms. So the next question of what can they do? So what is the potential or functional potential of those organisms? And for that, we'd be utilizing more of a shotgun metagenome sequencing approach, where we're sequencing all of the DNA present in a sample. So all bacterial, eukaryotic, fungi, viruses, archaea, everything in that sample. And that really allows us to look at species and strain level resolution and some functional profiling. But with that, in my opinion, outside of some research, it's still at the point where it's pretty cost prohibitive. It's very expensive. Computationally, it's challenging to do. And also in terms of sample type, there's challenges in low biomass samples or samples where we have a lot of host DNA. Can be challenging to sort through all of that data to figure out what the real story is. And then lastly, what are they doing? So the previous two showed us who's there, what can they do. The last kind of question we'd be interested in is what are they actually doing in the host? And so these would be some of your omics approaches that you might hear about. Providing insight into the functional activity of those organisms allows us to say those organisms are alive and they are functioning in the animal. Some of those are metatranscriptomics, so RNA sequencing of all gene expression, metaproteomics, and then metabolomics. So really, in an ideal world, we would combine 2 or 3 of these approaches to really get a true detailed view of what's going on with the microbiome. But I think today, largely, we're still at the point of focused on the who is there question, and oftentimes using 16S sequencing to accomplish that. And so I'm going to focus for the remainder of my slides on 16S specifically. And so what is the 16S gene? It's a gene that codes for the 30S subunit of the bacterial ribosome. And so it's ubiquitous in all bacteria and archaea. It's about 1,500 base pairs long, and in that are 8 conserved and 9 hypervariable regions. But again, as I talked about with amplicon sequencing, from this, it does not give us any insight on functional capacity of those organisms, just which organisms are there. And then there's also some limitations in terms of distinguishing species when their 16S gene is very similar. So for example, we cannot distinguish pathogenic versus commensal E. coli, which would be relevant in pigs. But the 16S gene does have the largest curated databases, which allow for simple and very cost-effective analysis on the back end. So this is just a plot of the 16S gene. Here on the bottom axis, we have just what base pairs. So again, it's about 1,500 base pairs long. And then on the y-axis is the amount of variability in those segments of a gene. And so here marked as V1 through V9 are the variable regions. And so when we sequence, we'll use primers that anchor to these low variable regions on either side of one or multiple variable regions. And then by doing that, we can use the variability in those variable regions to then distinguish different microbes. And so in our case, we've been utilizing the 4th variable region in our research. So outside of simply sequencing variable regions in the 16S gene, there have been in recent literature some newer approaches, amplicon still approaches that allow for increased resolution. They do come with added costs and computational issues. They don't have necessarily databases there, so it's more difficult from that standpoint. But I think as we continue to move forward, we may see some of these approaches becoming more common. So one of those being sequencing the full 16S gene, and so all 1,500 base pairs. And so by doing that, we use all of that variability to distinguish between microbes, and that gives us greater resolution. So we can get down to species and even strain level resolution with this approach. Then we can also use other marker genes. Instead of 16S, we could use protein coding genes, which would allow for some functional insight of what's going on, as well as greater resolution. And so one of those examples that's been published in literature recently is the RPOB gene, and that may be a good candidate moving forward. So now that I've kind of briefly touched on just generally some of the approaches we could take, and now focusing on 16S, we'll talk about, now that we've taken the samples and sequenced them, just kind of what does the analysis look like, and then what are some of the measures we're interested in looking at from those sequences. And so after sequencing, data is just output as raw reads of nucleic acids, like this line. Really doesn't mean a lot to us, right? And so from there, we would input those sequences into some specific software for processing and quality control. Some of those examples being Mother, Chyme, Data2. We're currently using Mother. And then from that, we would get a matrix, as shown to the right here, where we have organisms here as rows, samples as columns, and then counts, or the number of those organisms, of each organism present in each sample. And from there, those are what we're really going to use for the remainder of the analysis. But with that as part of the quality control process and those initial steps, one thing that's really important is controlling contamination. We know there's bacteria everywhere. And so contaminating samples and getting sequences that are not necessarily representative of what you sampled can be an issue. And so number one, it's important that we're extremely careful when we're collecting samples and processing. So for example, our group has done some collection on colostrum and sows. And so with that, it's extremely important we're collecting that aseptically. And so we're not getting contamination from, say, skin microbiome bacteria. But outside of just simple sample collection and lab processing, it's also important to sequence negative controls or blank samples when we do our DNA extraction and sequencing. And so we'll take all the reagents of the kits and PCR products and sequence those as well, because we know there is inherent DNA in those kits. And so by doing that, we can use statistical methods to then identify what contaminants are just from reagents, then use some software to then remove those from our analysis. And so an example is Decontam-NR. And authors that develop that program, I suggest that 6 negative controls is usually sufficient for most studies, but that we should do contamination identification separately if we have multiple sample types within a study. So if we have colostrum samples and fecal samples, we should be identifying contamination in those separately. So once we've kind of gone through the processing and quality control steps, the kind of last step of that process is assigning or identifying what microbes those are. And so we'll align those sequences to reference databases. So some examples are the Silva database and the ribosomal database project for 16S. We're currently using the Silva database in our PIG work. But then from that, we will identify the finest resolution possible, which would be considered, in our case, species level resolution. And we really have 2 options when doing that. And these are 2 terms that you're going to see in literature and hear about in presentations. And so I'm going to spend a few minutes explaining the differences to hopefully allow people to maybe better understand those differences between these 2. And so one option is amplicon sequence variants, where we're taking those sequences and identifying sequences that are exactly the same. And if those sequences are exactly the same, we would consider them the same species. The other option would be OTUs, or operational taxonomic units. And there we're clustering based on similarity. And so we're saying those sequences are similar enough that we're going to consider them the same species. And so in literature, you'll see ranges from 97 to 99% similar to be classified as the same OTU. We're currently using 99% similar. But at the end of the day, those both are accomplishing resolution at the genus and species level. To go into a little more detail on some of the differences of the 2. Again, quickly, OTUs are clustering method where we're looking at similarity of sequences. But because of that, because it's a clustering based on what sequences are present when you're analyzing those data, you can't compare OTUs from one study to OTUs in another study. And so we're limited in that sense. Additionally, it's possible that if species are similar enough in whatever region you are sequencing, they may be classified as the same OTU, even though they are the same species, different species. So there's some pros and cons with OTUs. For amplicon sequence variants, or ASV, it seems to be a more recent approach as we developed models to more appropriately identify error in sequencing. We've been able to become more confident using ASVs. Another term you might see for ASV is ESV, or exact sequence variant. And so just keep that in mind as you're reading or listening to presentations, that ASV and ESV are the same thing. And ASVs are really a denoising method where we're looking at identifying errors, removing errors, and then when sequences are exactly the same, considering those the same species. And because of that, it allows us to compare across studies. It's at the end of the day, both of these approaches are valid and accepted and widely shown in literature, but the choice of that analysis will impact your results. And so it's important to keep that in mind. This is just an example of what those data might look like after we've assigned taxonomies. We'll look at relative abundance of those different organisms. And so relative abundance being the percent of the sequences that were represented by those organisms. And so here at the genus level, we see the top 30 different genus, genre, and different sample locations within the animal. And then from there, we could do some statistical analysis to determine differences in presence of individual bacteria. So from there, we're also interested in, from a high level view, the diversity of those microbial communities. And there's really two forms of diversity, alpha diversity and beta diversity, that you'll hear about. So to start with alpha diversity is quantifying the microbial diversity within individual samples. So when we think about alpha diversity, we're concerned with richness, evenness, and then diversity. And so richness being the number of species within that sample, evenness being the similarity in the abundance of those species, where an uneven community, so if a community is dominated by one or two organisms, would be considered less functionally diverse than a community that is more even. And then at the end of the day, diversity, which is kind of the combination of richness and evenness. So just as an example to kind of illustrate what that means, just put together 3 different samples here. Where on the far left here, we have a community that has high richness. So there's 4 different organisms represented by different colors here, but low evenness because it's dominated by this red colored bacteria, where the other 3 are at low abundance. Here in the middle, we have a community that is low in richness. So there's only 2 different organisms here, red and blue, but then high evenness because there's 4 of each organism in the sample. Then on the far right, we have a sample that's high in richness and evenness, where there's 4 organisms all represented at the same abundance. So from that, there's a few indices that we use to kind of quantify that diversity. And so just to go touch on some of those that we'll typically look at. Shannon Diversity Index is an estimator of richness and evenness, but that index puts more weight on richness. Simpson's Diversity Index is kind of the inverse, where we're still looking at richness and evenness, but there's more weight on evenness. And then Chow-1 richness is estimating the true species richness of a sample, and that index tries to account for unobserved species that may be due to sampling. So typically we'll visualize diversity, alpha diversity, with just bar charts to look at differences. Here we have some of the different metrics across different sample locations. And we can also, again, run some statistical analysis to determine differences in whatever group we're interested in. For a second kind of form of diversity that we'll often measure is beta diversity. And beta diversity, where alpha diversity was measuring diversity within one sample, beta diversity is measuring diversity or differences between samples, so dissimilarity between samples. And there's a few different measurements that we can use for measuring dissimilarity. Those are listed here. I'm not necessarily going to go into the details of those, but are all valid approaches and have pros and cons. But by calculating dissimilarity of those communities, we can then use methods like PermaNova, which is very similar to ANOVA that we would normally use for statistics, just with different assumptions. And then also ordination methods. One that we might hear about in other areas as well is PCA, or in this case, principal coordinate analysis, or PCOA, where we're partitioning variants to look at clusters of samples. And then from there, we can kind of visualize based on our variables or groups that we're interested in what may be driving those clusters. And so here we see on the left side, there is clustering of samples. When we color those by age, it doesn't seem like that is driving the clustering, but as we color those by sample location, we see that it looks like that is the large driver of the differences in diversity caused between those samples. Then kind of lastly, in terms of 16S, I know I've talked about how we can't necessarily have a functional idea of what's happening with 16S. There are some approaches for predicting function based on what microbes are there. So complicated approaches, but generally we're by what organisms are there. We can use enzyme and pathway databases to then predict maybe what could be going on. And so again, it has all those same limitations that come with amplicon and 16S sequencing, where we're limited in resolution as well as distinguishing similar species, but may have some utility when we think about trying to take it a little bit of a step forward using amplicon sequencing. So it's just a quick summary of, we've talked about kind of the approaches that are possible, and then went into some detail about sequencing and analysis of amplicon sequencing. I'm just going to quickly summarize some of the advantages and disadvantages that I see. And so some of the advantages of 16S and amplicon sequencing is that it does do a really good job of giving you a high-level overview of the community composition and structure of those samples or of that organism. And so along with that, it's also a cost-effective and simple method. And so again, cost ranging from $10 to $50 a sample. And it also requires a low amount of sample because we don't need to worry about host DNA or those sorts of things, because we're only interested in one marker gene. And again, also have the largest databases behind it, allowing for simple alignment and analysis. But it's also important to recognize the disadvantages that come with it, with everything. And so there is, again, limited resolution with this analysis, where we can really only see genus and species level identification. Depending on some of the approaches you use, there may be limitations in terms of comparing studies. And then we also, if we're interested in bacteria and maybe viruses or in fungi, we would have to use different marker genes and sequence the samples multiple times to do that. And as with everything, it's important as we go through the process of designing studies or evaluating studies to recognize there is bias that comes along with microbiome studies, as with everything, where things like sample storage, extraction, kit contamination, PCR errors, or even just the software used for analysis can change your results. And so it's imperative that we're careful in our study design and mindful of quality control to limit the bias as best we can. So it's kind of the last future outlook of what we see microbiome research moving to. We see kind of bringing back this diagram that I introed things with of, we see that as technology advances and things become more cost effective, maybe a combination of these different approaches being used, potentially maybe amplicon sequencing, or that who is their question with what are they doing, or metabolomics type questions, to really get a full picture, detailed view of what the microbiome looks like and how it's being impacted by whatever management or treatment thing differences we're interested in. So with that, that's all I have, and I'll hand it back over to Dr. Greiner to talk about next time.
So thank you, Dalton, for finishing the series or the webinar presentation for today. As we look forward to the next series, which will be in June, we will be talking more about the pigs specifically. So we've outlined today a little bit, hopefully, to get you in the right mindset for the microbiome, what it is, where it is, some of the challenges with sampling, and then certainly getting a little bit more comfortable with some of the terminology and sequencing options that we have available. So next time when we meet, we're going to talk more about the microbiome itself, specifically in the pig, what we currently know about it, what gaps in knowledge are there, and what can we start doing with the information that we do have available, specifically to the animal of interest, which is, of course, our pig. So with that, today we are thankful for all of you that have stayed with us through our technical difficulties at the beginning. We're certainly happy to answer any questions through the chat today and in the near future as well as they come along.
All right, yeah, as Stacy put in the chat, feel free to add any questions now. I do have one to kind of get us started, but if anybody else wants to add one, you can go ahead and just put that into the chat feature, and then I'll go ahead and make sure we have Laura and Dalton address that. In your guys' reading or in your lit review, or maybe even some experience on your own, have you guys looked at how much variability there is? Let's say if you go in and do a rectal swab, let's say, if you took the same rectal, pig and you rectal swabbed them 3 times, how much difference was there? If you did the same process, as close to as you could, being as careful as possible, how much variability there was just in different samples. Have you come across any of that in your guys' research or in the literature?
So I can't say that I've necessarily read specific in literature or that we've actually done that to quantify variability, but I can say there definitely is variability regardless. If you were to take that approach, you would see differences, right? Because if we're taking, say you're extracting DNA from 1/4 gram of fecal sample, you can't possibly get every possible organism that's in that pig represented in that. And so there is some variability there. A lot of the techniques, or like I mentioned, some of the diversity measures do try to account for that. And so at some level, there's some estimations to try to account for unobserved things, but there is definitely some variability along with that, and probably most of it's inherent. And so that kind of comes back to doing everything we can to be consistent and careful with what we can control, with the understanding that there's inherent variability that we really can't.
Oh, very good. Yeah, it was definitely a very deep dive. Certainly appreciated the information. As you're looking at the different options, comparing short reads to long read sequencing, understanding the short read is maybe you can look a little bit wider, but you don't get maybe as deep compared to the long read. What would be some of the considerations that you would have to try to choose one method over the other?
I think in a very practical sense, right now, cost is the number one thing. 16S would be that short read sequence where we're just sequencing one or a couple of those variable regions. So you only need short sequences. You can do a lot of them. As we get longer, cost goes up substantially, and it gets harder to analyze that data. But depending on your question, right, if you are really concerned with getting more species and strain level information versus a whole broad overview, may be worth it.
Very good. All right, we're coming up on our hour. I don't see any additional questions in the chat feature. So once again, Dr. Greiner and Dalton, sure want to thank you for all your time and effort, and really looking forward to maybe the more applied methodology on the next one. I can appreciate certainly setting the stage for that one by doing a nice deep overview of microbiomes and the tools that we use to assess them. So very much appreciate that, everybody, and we'll look forward to seeing you next month.
Thank you.